Hello students, today we are moving to the last part of the chapter, that is strategies for the enhancement in food production, that is sericulture. Sericulture is actually the breeding and management of the insects for the production of silk. So there are numbers of arthropods there present who actually undergo the process of metamorphosis, and whenever it metamorphoses from one instar larva, from one stage of the larval stage to the next larval stage, then it produces some type of thread, some type of silk, and it form a cocoon within which they become adult. Okay, and that cocoon we can extract. We remove that organism which is present inside it, and after that, from that cocoon, actually from that one ball of silk, we can extract the threads, and we just processed it for the making of cloth. Okay, so first of all, just see what that various types of organisms, arthropods, are present, what type of organism they are for that sericulture. Okay, so what are the types of silk? See, there are two types of silk. One is called as mulberry silk. Mulberry is also the cultured silk, and the non-mulberry silk, which is also the wild silk. That means it is found in the wild tree. Now see, some type of organisms, they can be cultured and reared inside the home. Okay, inside the house, or inside, suppose actually inside one house where in the light, in the controlled environmental condition it can be grown, they will be known as the mulberry silk production. But sometimes also this is happening that some type of clothes, they are extract, some type of silks we can able to extract, but it is not possible that they can be collected, or they can be cultured inside the house. Okay, they one individual, if suppose they are thinking of producing the non-mulberry silk, where the cost is also very high, they need to have the proper one field. Okay, one complete paddy field where they need to have the tall trees like som or swallow, so that in that twigs or in the branches of the that tree, som or swallow or oak or castor or any type of plants which is the food plant of that specific arthropod which is producing the silk, in those twigs actually they complete the life cycle, and from that plant we are able to extract. We just collect the that silk ball, and after that we will just process it. Okay, so there are two complete different mechanism, there are complete two different setup for the sericulture. Mulberry silk is very easy to be cultured because it do not require that much of area, or that much of, suppose, long tall trees. Okay, then if it comes to the non-mulberry, then they have to have one complete paddy field with the tall trees that are basically the jackfruit or mango or som, swallow, castor, oak plant. In those plants they grow and complete their life cycle, so that we can able to extract the cocoon. Okay, so there are the two types of silk depending upon their availability or their process of culture. One is within home, another is outside. Okay, so one is mulberry, another is non-mulberry. Now next one, depending upon their life cycle. Okay, depending upon their life cycle, the silk also can be of three types. Okay, so see, suppose this is happening that one type of silk, that means we are talking about the silk organism. So that silk organism is completing its life cycle in only in suppose 1 year. Okay, so it produced the cocoon only once in their life, once in their year. Okay, suppose in the October month they form the cocoon and it become adult. Okay, then once again the adult lay eggs in February, March, form the instars up to June, July, August, and after that in September, October once again they form the cocoon. So in 1 year they produce only one set of, that means only one life cycle in 1 year. So that type of silkworms who complete their cycle in 1 year, then this type of organisms, they are known as univoltine. Okay, and second one is known as bivoltine. Bivoltine means it completes its life cycle twice in a year. Okay, so obviously the products will be high because we can able to extract two sets of cocoon from the two different time of the year, maybe on the March month or April month, and once again in October or the September month. So that is known as the bivoltine type of silk. Next one is the multivoltine, where we can extract the silk many times in a year. That means it completes the life cycle within suppose 3 months or 2 months. Okay, so that's why the life cycle will be increased in a year. So obviously their production will be also very much high, isn't that? So there are three types of silk depending upon their life cycle. If one life cycle completes in 1 year, that will be known as univoltine. If the life cycle completes in suppose in 2 times in a year, that will be known as bivoltine. If many times in 1 year, that will be known as multivoltine. Now after that, let's just move to the different types of silks. We have Dasar silk, then Muga silk, Eri silk, varieties of silks we have. Okay, so let's just see all of them. First of all, Dasar silk. So the scientific name of the species of the Dasar silk producing worm, that is known as Antheria mylitta. Okay, so just see that clothes we extract from the tassar, that is known as kosha. Okay, remember that. So this kosha is obtained from the oak forming larva. That means in the oak tree, the organism, that worm, they feed the oak plant, and after that they produce the cocoon. So this is the oak feeding larva, from where we can able to extract that type of cloth from the tassar silk, that is known as kosha. Next one, this tassar cocoon is attached to the twig of the sal tree. So there are different type of feeding plants are there. So what are they? Maybe the sal tree, oak tree, or some another type of trees also. So trees basically can be of two type: primary feeding plant and secondary feeding plant. So primary, which is actually basically desired by the worm, or their production or their growth will be maximum by getting that plant trees. And if suppose the organism they can also survive in some another type of plants. Okay, by taking that plants also they can survive, but it is not desirable or their growth is moderate, that type of plants will be known as secondary feeding plant. Okay, so this is the tassar silk. After that, let's just move to the eri and the Muga silk. Now after that, let us see the other type of silk also. The second silk, which is very much important, is the Muga silk. So Muga silk is the yellowish or golden colored glossy type of silk, which have very good tensile strength. Also its durability is very high, and also this one is actually is called as the superior quality silk. Okay, so what is the scientific name of the worm which is producing this that Muga silk? This is known as Antheria asama. Actually this is collectively cultured in only Assam. But if you see the other type of silk, like the other mulberry, then the eri, that type of silk, they can be produced by the other parts of India also, like the Bengal, then Bihar, UP, Karnataka, in those regions also, the people, they practice sericulture. But if it comes to Assam, in the normal natural climatic condition, naturally the Muga silkworm can be cultured, and the high quality silk it is producing, which also cost very high and it is exported to the different parts of the world, is the Muga silk, which is chiefly only found, can be reared in Assam in natural condition. So what is that? That is Antheria, the organism, that is Antheria assamensis, or simply Antheria assama. So what is this? This is the golden yellow in color and superior in superior silk, and it's very much superior to the Eri silk or some other mulberry silk, okay, in its gloss, strength, or in durability. Okay, so it's also very much excellent for embroidery. So this is the Muga silkworm. What are the plants? They are generally taken by the Muga silkworm. They are swarm or swallow. Remember that this Muga silkworm, it is chiefly found, collected, cultured in only Assam. Okay. Next one, see the another type of silk, that is the Eri silk. The scientific name of the species of Eri silk producing worm is Philosomia ricini. It feeds on the castor leaf, so its plant, the feeding plant is the castor. Okay, and it is reared indoors. Indoors means which can be cultivated inside the home. They do not require any large or huge area, paddy filled long trees. Within the home, they can be cultured. The cocoon is soft, yellowish and white, okay, who actually provide us a little warmth, not the type of silk or not the type of product which is used naturally, normally can be wet. But if it comes to the Muga silk, this Muga silk provide warmth during the winters, and it provide cold during the summers. Okay, so that type of silk Muga silk actually is. But if it comes to Eri silk, rather it is called as a silk, it actually provides us warmth. So it's just like the wool. Okay, so just like cotton, right? Next one, mulberry silk. Mulberry silk worm is known as Bombyx mori, where it feeds on the mulberry plant. Okay, so there are different varieties of silkworms. We need to know their scientific name. They are food plants, primary and secondary. They are not that important, but at least only one feeding plant you can remember. After that, let's just move to the proper procedure by which we can able to collect the silk from the different types of silkworms. Now after that, see its life cycle. So first of all, this life cycle of the mulberry plant will include the whole stages where the adult produce the egg form the egg hatch, and the caterpillar comes out. That caterpillar feeds, and after that it forms the larval stage. First is the larva, it metamorphoses to the second instar larva, then metamorphoses to the third instar larva, then metamorphose to the fourth instar larva. Okay, so this way they will form the four larvae. Then after that, the fourth larvae, they actually form the cocoon. Inside that, they completely become adult. Okay, and after that, the adult, by breaking the cocoon, it comes out. So that is the life cycle. Just see how it is. First of all, in this whole cycle, the male and the female, they can be attracted to each other by one hormone. Actually, this is called as the attractant for the sexual intercourse, that is the pheromone. The female release one pheromone that attracts the male and it leads to coitus. Then after that, the females, they will lay around 300 to 500 eggs. So from the 300 to 500 eggs, all of the eggs cannot proceed the life cycle because it will require the efficient numbers of the trees, the resources where they will include the where they will go on feeding, and after that it will produce the cocoon, right? So if we have the less food, less plants, then what we can go for, that some of the eggs can be stored, they can be preserved in cold temperature, and some of the eggs, they can go for the next life cycle. So just see, some of the eggs, they can be stored, but yes, the eggs what we are choosing for the starting of the life cycle, first of all, they have to be washed in 2% formalin. So formalin is actually one disinfectant. Maybe it is possible that some of the bacteria, virus or fungus, they can also present on the surface of the egg, right? So if the egg hatches, then it forms the caterpillar. That bacteria or the fungi can go to the body wall of the caterpillar. Then after that, it may hamper the caterpillar growth, can kill, and as the silk diseases, they are very much contagious. They completely ruin the whole actually culture. Okay, within days, within 2-3 days, they can all die. So that's why too much precautions are taken. Okay, too much actually precautions that can prevent the growth of the foreign particles. So that's why the eggs have to be washed with the disinfectant, and that disinfectant is formalin. So with 2% formalin, it will be washed. Then after that, the egg hatches within 10 days. Okay, so whenever the egg hatches within 10 days, normally what happens, the eggs are allowed to hatch in a bamboo tray, you can say. Okay, so in the bamboo tray, they will hatch and it will form the caterpillar. So that caterpillar, they are the worms, the baby worms, they are known as the caterpillar. Now this caterpillar, they are then transferred to the food plants. Okay, so whatever the food it will take, the caterpillars will take the food gregariously, and it will grow, attain its size, okay, attain growth in the food plants by feeding the trees, okay, by feeding the leaf of the tree. So what will happen, these caterpillars have to be transferred to the food plants in the twigs, in the branches, okay. Then after that, it will start to take the plant, okay, and after that it will attain growth. So whenever the caterpillar attains sufficient growth, then after that it metamorphoses. That means it leaves the outer skeleton, exoskeleton, it attain growth. Then after that it will start to feed once again. Okay, so this way, this process actually of growth and shedding their exoskeleton, what is that known as? I have given one term in first year itself, that is known as ecdysis. So ecdysis is a phenomenon or metamorphosis where one organism, one larval stage of the arthropoda, they actually metamorphose to the next larva by shedding their exoskeleton and attain growth. Okay, so this larva, they will metamorphose and it attains the next larval stage within 25 to 30 days. It forms the first larva, then it will attain the metamorphosis, then it forms the second instar larva, then once again it will undergo metamorphosis, form the third instar larva, then undergo metamorphosis, and it will form the last larval stage, that is fourth instar larva. So this way 4 metamorphosis it will complete it. Okay, so first of all, suppose this is the caterpillar, right? So first metamorphosis, okay, this is the first instar larva. So each of the larval stages, what we can call it, this is the instar larva. Then it will undergo second metamorphosis, then it form the second instar larva. Then undergo third metamorphosis, form the third insta larva, then undergo fourth metamorphosis and it will form the fourth insta larva, and it is the completely mature larval stage, right? So now what happens in the fourth insta larva, there are little changes occur in their body. Okay, so the first insta larva is very much small, around around 1 centimeter, 2 centimeters. So this way it will attain around this much growth. Okay, that means around 5-6 centimeter it will grow. Now what will happen, this fourth insta larva, suppose this is the larva. Okay, it will have the small eggs. Now what will happen, it will form two glands. Okay, so two glands will be formed on their segments. Okay, so they are the externally segmented animals, so segments will be visible from outside. So one segment will form the protein part, okay, which is actually present beneath in the salivary gland. They have the capacity to produce the saliva. They release the saliva, and whenever the saliva comes in contact with air, it actually become the thread like, and this protein, this saliva actually will be known as the fibroin. Okay, so see, the fourth insta larva form two glands. Okay, what are the two glands? See, the first gland is the salivary gland, so that salivary gland will produce the saliva. Okay, suppose this is the thread. Okay, and this thread, this is the cross section, so this thread will have the saliva. So this saliva protein part is known as the fibroin. Okay, now after that, just see, it formed a thread. That thread can be 1000 to 1200 meter long thread. Okay, so at a time it will produce that much long fiber. Now after that, just see what will be the function of the another gland. So that another gland, the other gland produce a gummy substance called sericin. So around the fibron there will be a layer. Okay, and that layer is known as sericin. Okay, that is one gummy substance, sericin. So that sericin is actually of no use. This fibroin is actually giving us that luster or that color or that shine, and if sericin is present, then this sericin actually will not going to give us that luster or that smoothness. There's that specific texture of that silk. Okay, so that's why what happened, one procedure is here, that is degumming procedure. So that gummy substance sericin is actually removed by boiling those cocoon in the alkaline solution, like normally in Assam's different villages. What type of alkaline solution they use? Normally with the khar, okay, maybe papaya or maybe the peel of the bananas, they use it. Okay, they just boil it in water. They use 2-3 of the papaya, and after that they boil. Then whenever it boils, this procedure is known as degumming procedure. And in this degumming boiling procedure, what happened, this sericin is removed and this fibroin is left, which is going to have this specific color, texture. Okay, so that is the fibroin. Okay, this fibroin is produced by the salivary gland, and another gland which is produced in the fourth insta larva, that will produce this gummy substance, that is called the sericin, which is present on the outer side, that actually makes the proper shape of the cocoon also, because this salivary gland is producing the thread. Suppose this is the worm, right? So around the worm it is producing this fibroin. So this fibroin around this worm, suppose this worm produces this fibroin, and after that it will start to move. Okay, so it will be like this. So whenever it is moving, these fibers, if they don't have any sticky substance, then how it will be attached to each other, isn't that? So that's why this sericin is also very much important, so that the fibers can be attached to each other and a cocoon can be formed. So this is the cocoon inside which the worm is still present, the fourth insta larva is still present. So obviously we should know that whenever it is forming cocoon, they cannot take food. It is completely a non-feeding stage. Okay. So just see, this caterpillar formed the cocoon and become the organism will not take the food. So it's the non-feeding stage. Now after molting, it forms the adult, and by breaking that cocoon it comes out. See, in this stage what happened, we will kill that fourth insta larva. Yeah, it's little inhuman, but yes, we have to kill it so that it cannot break that that fiber, okay, and it is allowed to come outside, okay. But very often nowadays it is also have some procedures by what, without killing that animal, without killing that larvae, we can able to extract the fiber, okay. So some of the organisms are also here that whenever they form the cocoon, they leave one very small opening. That opening is known as micropyle. So with that micropyle it can come out, but very often it breaks the mouth part of the cocoon and it comes out. So whenever it comes out, the threads, they will be breaking, so we have to join, and whenever we make that type of thread, that thread will not have that type of texture, because in different regions it will have different weight, okay. So that's why it has to be killed, okay. But sometimes for some of the worms, this is not done, that they have to be killed. For some of the worms, they are killed. How they will be killed? Whenever they form the cocoon, suppose it completes forming cocoon, then before it breaks the cocoon and come out and become the adult moth, okay, become the moth, what happened, what we are going to do, we are going to take the cocoon in the kitchen, and in the kitchen, suppose there are the firing burner, and just above that they will have the bamboo, some type of bamboo sheet, okay, and under that the cocoons have to be stayed. Cocoons are kept for 3-4 days, so within that heat some of the cocoon die, and if it is not, then it is boiled. So if it is boiled, then obviously within that cocoon the larva, fourth insta larva, they die. So what happened, that before it comes out, the cocoon are boiled so that it cannot break the thread, okay, and after that the threads are collected by degumming procedure. When after the degumming procedure, the cocoon, the fiber starts to come out, then after that it will be the next step, that is the reeling, okay, reeling and re-reeling. So in the reeling, around the bobbin, the fibers are allowed to form or make coil, and it will form the different varieties of size of the bobbins, like that, so that it can go for the cloth making procedure or that weaving procedure. So what happened, this after molting, this that fourth insta larva, which is metamorphosing to the adult, that adult will be known as the moth. The moth come out, and this way the life cycle completes, okay. So this is the life cycle of the silkworm. So that's all about you need to know. And after that, let's just move to the last part, that is some of the diseases of the silkworm. Let's just move. After that, let's just move to some of the diseases of the silkworm. So the silkworm can be affected by all the types of pathogen, like the bacteria, virus, then protozoa and fungus. So let's just see, first of all, the disease is pebrine, which is caused by a protozoa. You just need to remember, like P, pebrine, it's caused by protozoa, also starts with the letter P, okay. So the causative organism, that protozoa name is Nosema bombycis, okay. What are the symptoms? The brown dots appear in the worm, okay. Suppose this is the worm, in the body brown dots appears, and after that it unable to spin because it become restless, non-feeding, die, okay. So it will not able to form the cocoon. Next one, the next disease is muscardine. Muscardine is actually caused by one fungus, that is Spicalia botrytis, okay. What are the symptoms? It's actually a skin infection caused by the fungus. So in the skin, if they have the infection, then how they will shed the skin, right? So they will unable to mold, or they will it's not possible that they can undergo metamorphosis, and after that it dies. Okay, so these are the symptoms of the disease muscardine. The third disease, which is actually caused by one virus, and the fourth disease is also caused by the virus, names are not needed. One is Fletchery and another is Gracilli. Okay, so Fletchery and Gracilli, both of them, they are caused by the virus. So just see the symptom. The Fletchery, it become brown in color, fail to form the cocoon. Okay, so we cannot undergo cocoon formation and become adult. So this is the viral disease, Fletchery. In Gracilli, what happened? The skin becomes fragile and breaks easily. Okay, easily the skin breaks off. So obviously it cannot undergo molting. Metamorphosis cannot be possible. A white liquid type of thing comes out from their body. Okay, and after that it become restless, then become non-feeding and die within short period of time. Maybe within the next morning, if they suffer affected by the virus, in the next day they die. So these are the different type of diseases. Only 4 diseases you can remember, it's more than enough. So that's all about the disease. Hope you have understood sericulture. We have completed also the chapter strategies for the enhancement in food production. This chapter is small, very easy to remember also, but yes, easy to forget also. So that's why you have to see frequently, you have to study the NCERT also, write notes and see the video. So that's all about this chapter. In the next day, we'll see another chapter, that is microbes in the human welfare. So that's all about the chapter. Thank you.